Thank you so much, Pramila and John, for inviting me to this wonderful session. I like how diverse it is as far as levels and approaches. So as was mentioned, I am a member of a program called Autism BrainNet, supported by a variety of foundations, including the Simons Foundation. They largely support it now. And I have served as not only the sort of Northeast collection site for cases, but also as a neuropathologist across the whole program. So when a brain comes in and they need a neuropathology report, I provide that free of service. And it really, sort of gets at the question of, we've heard about behavioral deficits that are present in autism. Certainly these come out of the circuitry of the brain. We've also heard about other organ systems that show dysfunction, like the gut. And interestingly, in some of the previous talks in this broader session, we've heard about T-cells that actually collect in the gut in an elevated incidence in autism. So I was looking at these cases with this perspective. I generally study in my laboratory the circuits that underlie specific behavioral deficits, and we use genetics to do that. And, if you look at the sort of, this was a real wonderful review in Nature Medicine, and if you look at all the genetic causes of autism, this is a fairly large, example of them. There are ones which are found by exome sequencing. There are some that are copy number variations, deletions, or duplications. There are syndromic disorders that many people are familiar with. If you aggregate all those cases, you really only explain a very minor fraction of all of autism. These are a couple of the genetic things that we study in our laboratory, and we certainly use these tools to target specific brain circuits and really understand where sociability deficits come from, aggression comes from, things like that. But I was always intrigued by, this huge amount of autism that we have yet to explain, despite all this tremendous genetic information that has grown over time. And in fact, one of the things that led me into autism was this paper back in 2005 by the laboratory of Pardoe in Hopkins, where looking at the actual brain from individuals that have passed away with autism, the postmortem brain, they found a surprising thing, which is that there's an elevated innate immune response, including various cytokines, chemokines, and growth factors, which really argues for an ongoing innate immune activity in the brain, even in all ages. Really, I think they had a diversity of ages in these cases. This was in 2011 really seen again in this seminal paper from the Geschwin lab, where they found by transcripts analysis of transcripts in the brain, they found an elevated transcripts reflecting innate immune activation of astroglia and microglia in a subset of cases you can see here, and a reciprocal decrease in many of the neuronally important genes, including some that are involved in autism spectrum disorder. And they had an enrichment for immune response, as well as, interestingly, antigen processing and presenting. So that kind of gets to a little bit of an introduction before I get into the results that we uncovered, which is kind of, what about the adaptive immune system? So antigen processing cells will take a peptide antigen broken down from a virus, or sometimes it's tumor antigens, and those will be displayed on the surface of the cell, often a class 1 MHC molecule. And then what the adaptive immune system has is this tremendous diversity of T-cell receptors and antibodies, which that diversity is produced by somatic recombination, subject of Nobel Prize many years ago. But it really is the power of that system to recognize very specific epitopes. In the process of a T-cell, cytotoxic T-cell recognizing this, it will that binding will evoke release of perforin, which makes pores in the membrane and release granzyme that goes in and promotes destruction of the cell or cell processes. So that's the kind of schematic version. Here's an actual image where a T cell is attacking a tumor cell. So more recent Nobel Prize winning subjects were the fact that the immune system constantly surveys the body for cancer and contributes to limiting cancer through this process. And here you can see, instead of this kind of raggedy drawing, you can see what actually happens, which is there are these little Bleb-like structures that evaginate off the surface of the cell in response to this attack. And these have been variably called Blebs, and there's a specific biology that's involved in forming those Blebs. So I want you to remember the cytotoxic T cells and the Blebs and things like granzyme. Okay, the other information I want to give you is a little bit of microanatomy of the brain. So there is a blood vessel in the brain. It has to be supplied with oxygen and various nutrients. But T cells come from that lumen of those blood vessels, cross the blood-brain barrier, which are uniquely forming tight junctions in the endothelial cells in the brain. It gets into the perivascular space, the cerebral spinal fluid, and then to get into the brain itself, it has to cross this barrier called the glia limitans, which is formed by astrocyte foot processes. So just keep those structures in mind as well. So this is really the surprising initial observation that I just happened upon as I was signing out these autism brains. So control cases, here's an 1-year-old and a 25-year-old. Even control cases have some lymphocytes. This is the blood vessel lumen, this is the endothelial cells, this is the CSF space, and this is where the glia limitans would be. So you can see these little round dots, these dark dots, those are lymphocytes. And even controls have lymphocytes in the perivascular spaces at very low levels. But what was surprising is in the autism cases, you can see a very large increase in the number of these lymphocytes around the blood vessels. And we decided to systematically look at that. Marcello and I, he was a fellow in the pathology department working with me. And we photographed images just like these to find all the blood vessels affected by lymphocytes in controls and autism cases, making many photographs from all the slides. Then we did an automated counting of these, based on the morphology. You can see with your eye what is a lymphocyte. We used methods to automatically score those lymphocytes with some segmentation of the images. And we got these lymphocyte counts in the control cases, and these are the autism cases. And as you can see, there are individual blood vessels with very large numbers of lymphocytes. And the median value for the top 15 lymphocyte blood vessels across cases showed that autism was vastly exceeding, and as much as maybe 65% of cases, the control lymphocyte numbers. And here are there are actually 5 genetic things in our collection. One is P10 heterozygosity, which is a megalencephalic brain disorder, but P10 itself does regulate the immune system, and there's lymphoid hyperplasia often seen in that disorder. Also a cytogenetic abnormality where there's an X to a Y chromosome, XYY. Those are the 2 conditions where we did see these lymphocytes to be elevated. Where do the lymphocytes localize, and what are the ages and those kinds of features? Well, that's shown here. We saw elevations in males and females across gray matter, white matter, and leptomeninges, segregating out those vascular cuffs that we had shown. Interestingly, there was a trend towards a stronger signal in the females than males, which we wonder whether that might relate somehow to females being clinically presenting might require a higher load. That's just one speculation. We saw it in pretty much throughout the brain. It wasn't like the lymphocytes were attacking a very specific compartment of the brain. We could see these lymphocytic cuffs in the cortex, in the basal ganglia, the cerebellum, the thalamus, and various other places. So it wasn't region specific. And this is not so different from something like multiple sclerosis, where the lymphocytes and the demyelinating plaques do have some propensities, but they're quite variable in their locations. The lymphocyte was elevated across ages in red. And you can see the controls generally with age, the lymphocyte numbers seem to go down in our analysis, whereas they were trending up in the a little bit with age in the autism cases. So it really persists out until very late ages. Do you see that lymphocytic increase? It wasn't related to epilepsy. Some of the patients that we had did have epilepsy. In fact, we are a collection site for sudden unexpected death in epilepsy. So conveniently, we had cases that were epilepsy for other reasons. And we did not find any correlation of history of seizures versus no seizures in the lymphocyte numbers. Maybe a little bit of a trend in the gray matter, which may not be surprising, but it wasn't significant. And then if you segregated controls and cases, again, there's really no stronger signal in the history of seizure cases. This is some examples of immunohistochemistry to define the subtypes of lymphocytes. So lymphocytes are T cells or B cells, and then within the B cell categories, there's CD4 and CD8 cells. CD8 is the cytotoxic cell that I was describing earlier, and we stain for granzyme, which is one of the effector molecules of NK and T cells, natural killers and T cells. And what we found was that this was a T cell predominant immune reaction, far fewer B cells. And if you look at the whole slide scanning with a CD3 immunostaining, we could see an increase in autism relative to controls. So, sort of bypassing the vessel-specific imaging. And we did see some of the lymphocytes spilling out beyond the blood vessels. It wasn't solely in the blood vessels. They're predominantly the cytotoxic or CD8 positive subset relative to CD4, but in both cases, there are some B cells present and there are some CD4 positive cells present. And again, if you do whole slide scanning, CD8s are enriched in autism relative to controls. So it looks like a cytotoxic or CD8 positive T-cell process in approximately 65% of the autism cases. The other thing that was very intriguing as I was looking at cases is this phenomena here, which is this is again, the blood vessel, this is the brain parenchyma, this is the CSF space. And I was seeing these eosinophilic round structures in the perivascular space across many cases. I was surprised to find when I stained for glial fibroacidic protein, so this is an astrocyte marker, that they were derived from astrocytes. They are GFAP positive. And here is that foot process I was describing, and certainly the Blebs kind of appear to be coming off of that glia limitans. They marked with two other astrocyte markers, S100B and ALDH1L1. You can see the scattered astrocytes here and then that debris. So and here's a quantitative assessment of this sort of GFAP perivascular space, volume or area, sorry. And it's much greater in autism than controls. And I think that the most interesting piece of data that we really found was this, which is that the median GFAP debris area around the blood vessel, when plotted against the median lymphocyte count across cases, showed a statistically significant correlation where the more lymphocytes you have, the more of that GFAP debris, which suggests, doesn't prove that there may be a causal connection between lymphocytes generating this debris. And in our mind, this really resembled the Bleb that I was showing you earlier, which is a cellular response to an attack by a cytotoxic T-cell by a tumor. There are CD8 cells in the vicinity of the Blebs as well. So we really think that there's a perivascular astrocyte Bleb getting formed in correlation to lymphocyte numbers, as was seen here. co-localizing with cytotoxic T-cells in autism. And interestingly, if you look in the literature, there are at least 3 papers already reported of elevated GFAP in the cerebral spinal fluid in serum. Some of you may know that Alzheimer's and other neurodegenerative diseases, people are beginning to see markers of these diseases, multiple sclerosis as well, in the serum. So the stuff that gets in the CSF does eventually work its way into the blood through the venous system. And interestingly, at least in this one example here, they correlated the amount of GFAP to the childhood autism rating scale. So in my view, this is one of the peripheral markers that might reflect this pathology that we're beginning to find. In other disorders where there is a cytotoxic T-cell attack of a particular cell, like polymyositis, which is an inflammatory myopathy, people develop weakness over time. There's typically a damage of the myocyte, and there's a fibrotic scar happening in that location. And we did find evidence of this such scarring in the perivascular space. We saw elevated collagen, which I don't quantify here, but I think it's in the paper. We also see irregularities of the vessel wall, the glia limitans space, and you can see a widening of that perivascular space as well. And this was seen in very young subjects. This is just the Jagged index, which is kind of computer-based a line drawn between the 2 barriers, and then asking how irregular that is, that edge. And here is the perivascular white matter as well. So both of them were elevated. And if we use some of these other markers in addition to lymphocyte counts, we could actually get a really tremendous segregation of the autism cases from the control cases. And as you can see, a couple of the genetic autisms, these 3 did not show lymphocytes. And one of them was the 15q1113 duplication, Dup15q they call it, disorder, and a few other genetic things, whereas other genetic things did seem to have the lymphocytes. And it did make you wonder if, maybe some of the cases, the lymphocytes had regressed, but you could still see sort of this scarring or the damage of the tissue that might have resulted from the lymphocytes. We also saw damage at other CSF-brain barriers, including the pial surface here. You can see the astrocytic debris. You can see it along the ependymal surface. This is the ciliated ependymal cells. And you can see a scar here, astrocytic scar, and you can see buried ependymal canals here. And then finally, the pial surface where we were getting some of that damage, you could also see a loss of the normal smooth neural pill in this very most superficial layer 1, which is interestingly where a lot of the thalamocortical from the second order nuclei project. And certainly sensory evoked responses, as we saw in an earlier biomarker, could reflect a disruption of those synapses here. So just to summarize, we think that we have at least the beginning of evidence to put forward a new hypothesis, which is that in the individual with autism, there might be ongoing infiltration of T cells that are reacting to some epitope at the glia limitans, generating this sort of reaction of astrocyte Bleb formation. So this work, only a few people were involved in this particular work. Marcello D'Estasio, who's actually now at Yale in the pathology department. And we've sort of continued some of this work with others in this group, and many of the other people are involved in other projects in our laboratory. So I can open it up to questions for people.
Thank you so much, Matt. We've got quite a few questions that have come through. So the first, you mentioned that T cells were elevated. Were NK cells at normal or diminished levels in ASD cases?
We have not systematically looked at NK cells, but there were definitely some small round cells in the perivascular space that didn't seem to mark with CD3 or CD3 or CD20. So I do wonder if there may be some other lymphocyte subtypes like that.
Okay. Another question, do you know if these blebs, are they specific for ASD or have they been found in other neurodegenerative or neurodevelopmental conditions?
That was one of the interesting aspects I didn't mention. I sign out postmortem brains for all sorts of disorders, and I've never seen this pathologic biomarker before. So at least so far, it's unique to this condition. There are some other, conditions which one might find it if somebody systematically looked for it, but it's never been reported in any other condition.
Okay. Let's see what else we've got here. Do you know if there's any work looking at brain imaging techniques that may be able to detect these blebs in vivo? And do you think that if they are potentially, specific to ASD, could they be diagnostic?
I think that the closest thing we have right now to this diagnostic connection to the blebs is the GFAP and the CSF in the blood. Interestingly, in our paper in Annals of Neurology that was published in 2019 that reports this work, we did discuss some of these issues, and one of the things we found in the literature is that the subarachnoid space is expanded, the CSF space is expanded, and there are sort of individual reports of dilated perivascular spaces in autism. So we think that those might correlate to what we're seeing, particularly the subarachnoid space expansion could relate to some impairment in drainage of fluid through the arachnoid granulations if there's debris that's somehow plugging that's generated through this inflammatory reaction. The blebs themselves, I think maybe below the resolution of imaging, but maybe with some nuclear medicine approaches targeting specific epitopes that might become possible.
Given, since you said that there may be some CSF clearage issues, given the role or presumed role of CSF and maintenance of debris and sleep being so important for maintenance of proper clearance, what role do you think sleep or abnormal sleep patterns may have here in these findings?
Very interesting. Yeah, that's been Alzheimer's beta amyloid made clear during the sleeping period. And there's certainly a robust sleep phenotype in a number of individuals with autism. The question is that due to a defect in some of the sleep controlling circuitry of the brain, which we know, there are various circuitries that promote sociability, that might promote irritability type behaviors, things like that, repetitive behaviors, which are dysfunctional. So it could be in just that general class of circuit dysfunction that's causing the sleep problems. But I do think that one should wonder about that possibility that there may be some interaction between the two.
Okay. Still got a few more questions, so bear with me. Let's see. What is your theory on the role of inflammation in the onset of ASD? And do you think inflammation is a consequence or is it potentially causal?
I think that the evidence is pretty strong that there's ongoing inflammation even within the brain itself in individuals with autism. And we believe, at least in our current working model, and certainly to be refuted by further experiments if possible, is that, why is there an ongoing inflammatory reaction in the brain? We think that it might be very analogous to other T-cell based autoimmune processes in skeletal muscle, and as I mentioned, polymyositis as an example, or any other organ might be the driving force for this ongoing inflammatory reaction. The other question is this inflammatory response, which is not circuit targeted, how might you get the behavioral deficits there? And we sort of, it could be that this is happening even in utero and that maternal immunity might be, and we're going to hear talks about that next, I think. And it could be that derails some kind of epigenetics for the brain circuitry. But I think that we have at least some evidence now to imagine that there might be an ongoing kind of autoimmune process in the brain in autism.
Okay. Given that that may be the case, what sort of role do you think there could be for treatments that affect the immune system, such as IVIG?
I've certainly looked through the literature, and there's really high dose IVIG has in some cases shown some improvement. It's quite intriguing how that we discussed last session about gut leakiness, and then it reminds me of the fact that one particular epitope that goes into the gut is coming from the milk, which is folate receptor alpha. And there was some evidence in some of those cases that were presented in this broader session that reducing or eliminating the folate receptor alpha in the milk from their diet had improved some symptoms in some individuals. That's quite intriguing. There are steroid therapy papers out there, just very small numbers of those. So I do feel that we need to build further evidence for this mechanism by finding biomarkers in the CSF and in the blood. And as we do so, trying to get a therapeutic response in the levels of those, and then in parallel monitoring looking for some behavioral improvements. It would be a useful direction to go now.
A couple more. We got time for maybe one or two more questions. We won't get through all of them, but if you might want to take a look and respond offline. Let's see. Is GFAP a parameter in CSF testing, such as Mayo Clinic CSF panels? I guess.
I have not really noticed. Yeah, it's not. I don't think that it's a routine test that you can sort of send out for at the moment. We've certainly thought about making such services available in our pathology department, but I think that it's extremely important in the future to make that something that's readily available. And I think that it's really important to find additional markers that might be coming from those blebs.
Okay. Last question. Are these T-cell abnormalities likely due to any early infection, and how might they be linked to the maternal immune activation? I guess you talked about that a little bit, but if you could go a little bit further.
Okay, so viral infection certainly will trigger a histology that can look a little bit like this. It depends on the type of viral infection. And that was in our differential. We stained for some of the more common traditional viral infections of the CNS, and we didn't see marking for any of these, but it could certainly been one that is not more traditional. I think that that's in the differential of such lesions. And over time, I think that, maybe that will be explored in further depth. The other possibility is something that we know to be true, which is that people that get herpes simplex encephalitis will sometimes, following that infection, have a delayed recurrence of this encephalitis. And in those cases, it's sterile. So it's really just tipped you into what we presume to be an autoimmune process. So that's another model by which this could occur, in theory.